3, 2, 1, go. Greetings, my fellow biologists. Welcome back for another lesson with Miss Garcia. What we've been discussing so far when it comes to Unit 2 is cell theory, the different types of cells, prokaryotic, eukaryotic cells, and how those cells are capable of reproducing. In this lesson, we're going to hone in on those processes a little bit and focus on how cells actually go about creating the next generation of cells. And this is very important in eukaryotic organisms like ourselves. So let's pull out our guided note packet, a pen or a pencil, and we are going to learn about the cell cycle and mitosis. When cells divide, their main goal is to produce new cells. These new cells need a set of instructions for what to do once they are created. That cannot be done without DNA. DNA provides the instructions for everything. Now, naturally, in eukaryotic organisms, our DNA exists inside this structure known as the nucleus. We oftentimes think of DNA in its packaged form, which is chromosomes, but inside of the nucleus, DNA is actually just floating around in its unpackaged form, the balled up string that we discussed in previous lessons. That big string ends up condensing into chromosomes. But we also undergo cell division. We don't want our DNA to get tangled and damaged, so that's why DNA condenses into those chromosomes. This slide is meant to reinforce the idea that during cell division, DNA, which carries our genetic information, bundles itself up into these packages called chromosomes inside of the nucleus. At this time, please pause your device and label the structure that you see on your screen in your guided note packet. Before we get into the steps or processes that make up cell division, we need to first understand why cells divide. There are three main reasons why cells need to divide: growth, repair, and replacement. As we mature and develop, we require more cells. We do not have the same amount of cells that we had when we were toddlers. If you ever notice when you get a cut over time, that cut may disappear because your cells undergo division to fill in that cut. Cells do get old, and when they get old, they need a way of being replaced. We don't have a bunch of old cells just floating around in our body. Cells have an advanced mechanism by which they regenerate new cells, and we'll talk about that a little more later on. Now, there's different processes by which cells reproduce and divide. We are focusing on mitosis within this lecture. We'll learn more about meiosis in our next lesson. We've already talked about some of the things that make up binary fission. Now, before we get into the more intricate steps that make up the cell cycle, we just need to know for right now, the cell cycle is a series of events that as the cell grows, prepares for division, and divides into two new cells, it is comprised of three stages. And we'll talk about each stage in more detail, but those stages are interphase, mitosis, and cytokinesis. Before a cell undergoes mitosis, it needs to make sure it has everything it needs, and this happens during interphase. Interphase is a period of cell growth before the cell divides. So there are multiple steps that make up interphase. We have our G1 phase, which is the cell grows. Remember, this cell has to split into two new versions of itself, two identical versions of itself at the end of this process, and it needs to grow in order to do that. So then we have our S phase, and this is where our DNA replication happens because we do need DNA for those new cells. Then we're going to grow some more. So after all of that's been finished, interphase is ending, and then we can go into our next phase of cell division, which is mitosis. As previously mentioned, after our cells undergo interphase, they are then ready to undergo mitosis. Mitosis is where the division of the nucleus is going to happen, and it is most common form of cell division in somatic eukaryotic cells. And remember, somatic cells are the cells that are not our sex cells. They are not gamete cells. I like to remember mitosis as we're walking, you stub your toe on your bed frame. You are in so much pain, and you yell out, "Oh, my toe, sis!" We are repairing our cells that have nothing to do with sex cells, but we will touch base on everything that involves meiosis at a later time. I want you to understand that when mitosis is complete, we have two genetically identical cells. that are being produced, and these are called our daughter cells. As we go through the different stages of mitosis, please make sure you write down the bullet points that you see in each stage under the important events in your guided note packets. The first stage of mitosis is known as prophase. During prophase, DNA condenses and chromosomes become visible, centrals and spindle fibers appear, and we'll talk about their function a little later, and the nucleus begins to disappear. The next stage of mitosis is called metaphase. In metaphase, centromeres of the duplicated chromosomes line up across the center of the cell. So remember, chromosomes have duplicated themselves at this time, and so the spindle fibers which are forming from the centrals attach to the centromeres of the duplicated chromosomes that have lined up in the middle of the cell. So remember, chromosomes were duplicated during interphase so that at this point the two copies are going to separate, and we'll talk about how they separate within the next phase of mitosis. The third stage of mitosis is called anaphase, and it is during anaphase that the chromosomes separate and move along the spindle fibers to the opposite end of the cell. So the spindle fibers are pulling the chromosomes apart to opposite ends of the cell. Now, each set of genetic information is going to be concentrated around the centrals. And remember, the spindle fibers are branching out from the centrals, so we're pulling those chromosomes back to their separate sides now. The fourth stage of mitosis is called telophase. During telophase, two new nuclear envelopes begin to form, and the nuclear envelope is what's going to be protecting the DNA, which is held inside of the nucleus. And each of these nuclei contain the genetic information or the DNA for the new cell, and the chromosome will start to uncoil, exposing the DNA as well. The final step of cell division is cytokinesis. Now, this differs a little bit in animals and in plant cells, but nevertheless, the result is the same. In animal cells, the cell membrane is going to be drawn inward by the contractile ring until the cytoplasm is pinched into nearly 2 equal parts. Now, remember, the cytoplasm is that jelly-like substance that holds all of our organelles, so the contractile ring is going to contract until it pinches itself, and then we get 2 new cells. With plant cells, however, a cell plate forms halfway between the divided nuclei, and the cell plate splits the membrane that separates the daughter cells. Now, the process of cell division is a pretty complex process, and our cells do a pretty good job at making sure this goes flawlessly. However, there can be hiccups occasionally, and typically, when a cell is damaged, it will go through cell suicide or apoptosis. A cancer cell is mutated and out of control mitosis until a tumor is formed or an uncontrolled growth. As previously mentioned, cancer is a disorder in which the body cells lose the ability to control or regulate their growth. Now, in typical cells, when the cell is damaged or mutated, the cell will fix itself and correct that through cell suicide or apoptosis. However, if a cell does not pick up anything that is wrong, or it can keep on replicating that cell, and that cell can continue to undergo different mutations, and that can lead to uncontrollable cell growth, which is known as tumors, and tumors are cancerous. Okay, guys, that is mitosis. Thank you so much for being patient and taking the time to fill in your guided notes. Please remember that at the end of your guided notes, there is a practice activity that you will have to color in different images and structures related to mitosis. If you need coloring materials, please feel free to stop by Lab 6 after school. Thank you so much. Have a great day.